Hi there, and welcome to a video outlining cell division. So cell division has a distinct, really the two distinct pieces are mitosis and cytokinesis. I've included some other parts of the cell cycle just to highlight those. So the first part is interphase, and this is where cells typically exist until they're signaled to divide and make new copies. So you have gap 1, which is a growth phase, and some cells stay in gap 1. They're just kind of the growth, normal daily activity, so nerve cells, for example, stay here. But then there may be a signal that you need to make more cells. So epithelial cells, for example, that line your gut and your skin, those get that signal quite frequently. And so then you need to duplicate the genetic information. So I've drawn here a 4 chromosome type cell. So I've got the 4 here in the middle. Remember that humans have 46 chromosomes or 23 pairs. I'm just trying to keep things simple here. So you need to actually double that. And so what I've shown here is this doubling over as you get into prophase. I didn't draw it here in interphase, but you would be doubling that in the synthesis phase. And then gap 2 is kind of the last sort of getting ready for mitosis. So maybe you're making more chloroplast, mitochondria, so that every half, these 2 daughter cells you're going to have at the end gets what it needs. So the first phase, prophase, has these, now you have sister chromatids, which I've outlined in a different video. And so instead of just the 4 that we had here, now we have kind of the 4 pairs. And remember, they're kind of bound together by the centromere there at the middle. And you need to form these spindle fibers that are there. And the spindle fibers are going to ultimately attach to the sister chromatids. You also need to form centrioles. Those are kind of your poles that are going to allow the spindle fibers to have a pole to attach to and pull apart these sister chromatids. Now with the metaphase piece of it, now you need to very carefully make sure that these chromatids, sister chromatids, are lined up in the middle. This is an important checkpoint phase because you don't want to proceed to anaphase where you're pulling apart the sister chromatids until they are equally lined up, because you want to make sure that the end cell gets the same number, the 2 end cells that you started with. If you don't have that done properly, and we see this a lot in cancer cells where the cell division is, not really following the checkpoints and maybe is too rapid, you actually can get cells that have too many or too few. It's called hyper or hypodiploidy, and those are patterns that we can find in certain cancers, and sometimes we even use those to predict treatments and outcomes, depending on whether they have too many or too few chromosomes. So sister chromatids are lined up here. You need to have each spindle fiber attaching at the kinetochore. Okay, that's an important kind of pause point. So this part's a bit of a slower part. Prophase is also fairly slow. Now anaphase and telophase are fairly quick. Anaphase is where you quickly start pulling apart those sister chromatids from each other. So these are now going to eventually inhabit their own space in the new daughter cells. You also see some fibers here that don't really seem to be doing anything. They're not going anywhere. Those are actually fibers that push the cell apart. So not only are you pulling the pieces that you want, the sister chromatids, but you need to kind of spread it out into 2 sides. You make it a little bit wider so that you're pushing it apart so that you can eventually divide it up. Otherwise, everything's kind of still hanging out in the middle there. So anaphase will then proceed into telophase. In telophase, you'll get the nuclear membrane start to reappear. Hopefully you've done this correctly, and you've got your 4 chromosomes on each side of the cell. And that nucleus is going to eventually kind of fill in nuclear membrane so that we'll have a nucleus here at the bitter end. There we go, a little bit better, filling that in. So by the time we get to the daughter cells, they'll have a nice nucleus there in the middle. I'm not drawing all the organelles, but you assume there's, kind of randomly a fairly equal split of organelles. Of what would happen if one half the cell didn't get its mitochondria, it would probably be in trouble and wouldn't survive. But the hope is that there's enough of each type of organelle, they each get some to get started. So telophase is that sort of kind of reforming the pieces that are going to be a new cell. Now in animal cells, they'll actually kind of spread out a little bit and then kind of just pinch off in the middle. Plant cells, it's a little more difficult, and I focused on animal cell mitosis and cytokinesis here. But in the animal cells, you actually would have to lay down what's called a cell plate or a cell wall in the middle. In In animal cells, they have what's called a cleavage furrow. Literally just pinches off because it has that soft cell membrane. But the idea is that you have these two daughter cells at the end that are just like the parent cell. And we would want that, right? If we're making more skin cells, we want them to be like the skin cells we started with. We don't want them to be genetically different and not be behaving properly and functioning properly. Same is true of heart cells, liver cells, all those sorts of things. So the last part is cytokinesis, and that's where you have that kind of final pinching in two. And so sometimes you see that collected with mitosis, sometimes separate, but they are a linked set of processes. And keep in mind, as I draw these distinctive processes, it seems like I'm kind of jumping from here to here to here to here to here. You know, it's like if we looked at, you know, ages in humans, and we looked at, okay, here's this, you know, puberty phase, and here's, you know, infancy phase and adult phase. It's a slow transition, right? So we can't necessarily pinpoint it started on this day or that day. You know, it's kind of slow transition. So keep that in mind as you go through mitosis as well. So we're showing kind of distinctive identifying parts of those phases, but it's a slow transition. And some texts will even put intermediate phases like between prophase and metaphase. There's something called prometaphase that kind of shows how we get from here to here, kind of in between that. Keep in mind that the kinetochores, which I've outlined in a different video, are very, very key in this process, and that spindle fiber attachment is pretty key. Other than that, we're looking at duplicating, pulling apart, and then separating the two cells into two new daughter cells. That's all I have for the cell division process. Remember, the interphase is not part of mitosis, but it does get ready for mitosis, so I've tried to make those separate. Please contact me with any questions